So our next speaker is Dr. Lindsay Marshall. She's a science advisor to the Humane Society International. Dr. Marshall received her Ph.D. in cell biology and protein biochemistry at Dundee Medical School in Scotland. Her postdoctoral studies were primarily focused on investigating the inflammatory responses, repair processes, and innate immunity of the human airway. Throughout her academic career at Aston University in Birmingham, she continued this theme of human respiratory deference. And then Dr. Marshall left academia in 2016 and is now, as I mentioned, a science advisor for the Humane Society of the United States. And her overarching goal is to in this post is the encouragement of animal replacement in biomedical research. As we were putting this program together, we also came across one of Dr. Marshall's recent publications where she was discussing really the poor translatability of therapeutic research to human. And so, again, I think this is a very relevant topic for today's discussion. So with that, the title of Dr. Marshall's presentation is Replicating the Human Airways in Vitro from Normal Physiology to Disease Models for Drug Testing.
Thank you so much. Thank you for the invitation. I really appreciate it. And I love the work that Chris is doing. I think it's really important and it's really lovely to be part of the panel discussion. I'm looking forward to that later. I do feel like I'm going to change gear somewhat, and my disclosure here is that I'm not a safety scientist. So as Norb told you, I'm a biologist. So what I'm going to look at is more a high-level view of respiratory physiology and how are we modeling that and how might we model it more effectively, I guess, without using animals. And that, how do I move my slides on? Oh, there we go, sorry. Is because, as Norb said, I work for the Humane Society of the United States now, which is an animal protection organization. And you can hopefully see the picture of the dog here in the cage, which is just an illustration of why we do what we do. So this was a dog that was being used to get data for pesticide registration. And then together with my colleagues at Humane Society International, we managed to get the regulators to waive the requirement for these data. And I can't go into the details for that because that wasn't my project, but it did mean that we got the dog out. And it's not just about getting that one dog out. It's really, as I'm a science advisor with HSUS, my role is really to promote the best science. And we feel that the best science is often human biology based if we're interested in humans. And therefore we want to accelerate that shift away from using the live animals towards developing more NAMs, towards and NAMs, I should qualify, or what we would call non-animal new approach methodologies. I feel like everyone has a slightly different definition, but ours are very firmly rooted in that non-animal basis. So how can we promote the development of more NAMs for testing, for biomedical research, but also for safety testing and for chemicals and cosmetics and wherever else you'd like to use them? So what I'm going to talk to you about then is respiratory system, challenges and opportunities in modelling that. I've got 4 disease case studies that are very high level overview of some of what we had in that paper. And I'm more than happy to share that paper that Norb talked about if you'd like to go into this in more detail. And then I thought it might be helpful to have a look at where NAMs are currently being used for respiratory disease and where we might use them beyond that. So I hope you find it useful. So I feel like everyone says their organ system is complicated. So I'm going to say that my organ system is complicated. Human airways are the most complicated. They are a complex, complicated system made up of probably more than 41 different cell types. There are different composition of each of these different cell types in the different regions. The different regions have different functions. They're not just about breathing. I'm not trying to big them up here, but there's also a very important defense function in your airways, and they form this functional barrier. So because they're epithelial, they're protecting you. They're keeping your insides in and the outside out. So we need to be able to have an arsenal of defenses that's going to make sure that we're adequately protected from anything that we might breathe in. And if we think about, well, that complicated system, then we understand that modeling them is going to be just as complicated. So I've got some of the kind of, I don't know, buzzwords about what the airways look like really for human airways in here, so that you can see that actually we can't take all of these features and sort of scrush them all up together and turn them into something that would fit on the palm of my hand and make it into an in vitro model. We just can't even think about modeling the complexity of that entire airway system. So what we have to do is step back and model the functional unit. So we want to make this as simple as we possibly can, but as complicated as we need. So if we can get down to one cell type, let's do it. But if we need more than that, because we know that cellular cooperation is really important in airways, then we need to build up that complexity. So it's really about what model we need to answer the question that we're asking. And I guess I'm simplifying it here because I'm talking about biomedical research. So we're talking about something that's maybe more amenable to this flexibility than I know the constraints that you're under for safety testing. It's easy for me to stand here and say, oh, this is very simple, but that's kind of where we are for biomedical research, I think. And it's important that we think about the questions that we're asking and how we're asking them, because Thomas Hartung of Johns Hopkins University says this famous phrase that we are not 70-kilogram rats. And that it feels to me particularly true when we look at airways, because if we look at rodents, they are obligate nose breathers. Humans are not. They have a very different branching pattern to their airways, which is kind of crucial if we want to look at particle or drug deposition. Then that pattern alters the airflow and will alter where particles will land. Rodents have different cell types and different cell numbers, so there's different composition throughout those different functional regions of their airways. Rodents have a massively different immune response. That's important for everything, I think, but for the airways, what we find is that people are quite sensitive to having bacterial products in their airways because, you know, we don't really want them in there, whereas rodents, scurrying about on the floor, lots of bacteria in there, not so bothered, wouldn't mount such a robust immune response. So there are these significant differences in how we deal with invaders and respond to invaders. And I think we saw that quite exquisitely in the past few years with SARS-CoV-2, where we found that mice are not susceptible to that at all, and humans very definitely are. And this is not about shifting species. So we can look at different species for modeling respiratory diseases, and many different species have been used. And what we found actually is that using a different animal model might recreate a different aspect of the disease, but none of these animals give us the whole disease as we see it in humans. So we need to kind of think beyond, well, let's pick out a symptom in an animal and see if it models it. And I think that's where the NAMs maybe give us the power to look a bit more closely at these symptoms in a human relevant system. Animals are still used for biomedical research looking at respiratory disease, and these data come from the European Union, so they have a lovely database that has very beautiful granular data. And for each year, there are 4 bars. The first 2 bars refer to animal use for basic research, so that's looking at fundamental knowledge of respiratory system, respiratory function. And the second 2 bars are animal use for translational and applied respiratory research, so more disease-related research. For each of those 2, the first larger bar gives you the number of all animals, and the second bar gives you the number of mice that are used. And I use these data for 2 reasons, really. One is to illustrate how mice bear the brunt of this research. Despite those differences that we've already kind of flagged up that we're aware of, mice are used really commonly for this kind of research. But secondly, and more positively, I'd like to have a more positive message, we can see that the number of animals is reducing over time. I mean, we'd like it to catapult towards 0 really quickly, but it's really good that it appears that we're reducing our reliance on animals for respiratory research, and hopefully that's signifying a shift towards non-animal methods, because respiratory research certainly hasn't stopped. So I'd like to now just look at 4 different diseases, so human diseases, and take a look at the disease features and then the animal models that have been used, and just give you a few examples here of how we can do differently than animal models to be able to understand these diseases more effectively, I guess. So the first is what I did my PhD on, which is cystic fibrosis, which is a rare disease, so affects around 40,000 people in the US. It is a monogenic disorder in that there are mutations in one gene, but that is a modifier gene, so it has impact on other genes. So it's a bit more complicated than that. It's a multiple organ disease, and really the main consequence of it is a lack of hydration of secretions, which in the lungs means that you get this thick, sticky mucus secreted, which invites infection with opportunistic infection pathogens. So these are pathogens that wouldn't really bother a person that didn't have CF, but for somebody with CF, they colonize the lungs and it's really difficult to get rid of them. There's a hyperinflammatory response, which means we get neutrophils coming into the lungs, and those neutrophils are a bit overexcited and create a lot of damage, and there's no way to stop that cycle because the infection continues and is never really cleared. When we think about animal models, then there are various different types of animals that have been used. The mice initially, once the gene was discovered in the late 80s, there was, you can imagine, a boom in putting that gene into mice so that we could see, can we develop a mouse model? Mice don't get the disease naturally, so we have to do some sort of manipulation to drive the production of the disease in the animals. And in the mice, at least, there's no spontaneous lung disease. So you can give them exactly the same mutation as people with cystic fibrosis have, and the mice, their lungs are fine. In pigs, the newborns do tend to be born with lung infection, but what happens there is we see the sort of the impact of that exquisite host pathogen sensitivity that we have. So the pigs, newborn pigs, will get an infection, but it's with a different pathogen than a newborn baby with cystic fibrosis gets. So it's still kind of hard to translate, well, what do we treat our pig with? We can't put that into a human. So we still have this issue that we're not mapping our animal model onto our human. Cystic fibrosis, though, is a really great example of where we can use in vitro platforms, because there's a Vertex, or a drug discovery company based in Boston, that found these fantastic channel modifier drugs that have been absolute game changers for people with cystic fibrosis, because they can alter the activity of that mutated channel to make it functional again and address the issue with the secretions, that also improves lung function and prevents the infections. And why I mention that is because that efficacy data had to come from an in vitro platform. Remember, the mice don't get lung disease, so we can't show that a channel modifier will address lung disease in a mouse that doesn't have a lung disease. So whilst the safety data for these drugs came from animals, the efficacy data were driven from this in vitro platform using human bronchial epithelial cells. My next example is chronic obstructive pulmonary disease, or COPD, which is much more prevalent, affects around 13 million people in the US alone. And COPD is actually an umbrella term for various different respiratory conditions that all come together to mean that you get this irreversible airways remodeling, that mean that your lungs get a bit stiffer, they don't really flex as well, you don't really have that air capacity that you used to have, basically. And this disease is progressive, and it doesn't resolve when you stop the insult, which tends to be cigarette smoking, but it doesn't have to be. For the animals, so rodents are our friend again here with guinea pigs and hamsters, we need to either expose these animals chronically to toxicants such as cigarette smoke, or put enzymes or something like bleomycin directly into their lungs to drive that sort of emphysema. And what's really interesting about the animal models is if you stop that insult, the lung disease will resolve. So they'll improve. We don't see that with people. The disease might stop progressing if you stop smoking, but you won't get back to your healthy, pink, beautiful lungs. And I think I'll mention this again in a moment, but I think that might be due to partly what we're using to model our disease are young, healthy, often male animals, no comorbidities, nothing else kind of wrong with them. So we're not really mimicking our patient population with these animals. And there's no cure for COPD. If we look at asthma, we all know what asthma is. We probably all know someone that's affected by asthma, and that now we're looking at something that has an allergic component. We get an eosinophilic infiltration into the lungs quite often, and irreversible airways remodeling again. So this irreversible nature of the condition that is progressive. We get fibroblasts moving in, and those are the cells that give you scar tissue, so they're stiffer. So again, it means that your lungs are less flexible, and we get that bronchial hyperreactivity as well. So you get the spasms in your airways that are so frightening if you have asthma. We've got rodents again, if you look at animal models. Guinea pigs tend to have the airways hyperreactivity, so they're used to try and model that. And Drosophila, interestingly, so these are fruit flies that are used because of similarities in the genes and their innate immune response. So trying to tease out that immune element to this disease, we can use something like Drosophila. because they're small and they breed really easy and they're kind of cheap and they age really quickly. But again, if we want to develop the disease, we've got to expose these animals to something. So mice tend to be injected in their bellies with a protein of albumin that will drive the production of that respiratory issue. And again, when we stop the challenge, we stop the kind of disease. So we're not really modeling that long-term, lifelong condition that we see in people. And then my final example that needs no introduction is SARS-CoV-2, which we all know was this horrific, highly contagious respiratory virus, which we're still living with, which had disease phenotypes from mild to severe, with greater severity depending on comorbidities such as age or obesity or other conditions like diabetes. When we modelled this in animals, the popular choice were mice, hamsters, or macaques, so the non-human primates. Again, for the mice, there's no natural susceptibility, so we had to transfect the receptor into the mice so that we could drive that the infection. Hamsters, I think, are more susceptible. Macaques are susceptible, but only to mild disease. So it's kind of interesting that the macaques didn't get the severe lung lesions that the people did, despite that close genetic similarity that we have with the non-human primates. And again, I think my point here is that we understood fairly quickly with SARS-CoV-2 that those comorbidities played such an important role in how the disease might progress. And yet it's really difficult to model that in animals. It's really difficult to get one disease model, never mind trying to put 2 or 3 together. So we haven't got the ability to look at the different comorbidities. My positive take from SARS-CoV-2, if you'll bear with me about a positivity out of this, is that we got really good at sharing data so that when that genome was sequenced, it feels like everybody knows what the gene sequence was. So we could all look to pick out what would be the most antigenic region of this, what should we target, what can we put in our vaccine, so that we could really accelerate how we get something out there as quickly as possible. And also the success with repurposed drugs, which is where some of the NAMs have come into their own, which I'll talk about in a minute or two. So if we're thinking about respiratory disease research, then there are various examples of NAMs. I'm not going to go through all of these in a lot of detail because I don't want to bore you to death, and I don't think I've got time to. But we've got organoids tend to be stem cell based, so it recreates some of the architecture of the organ, whereas spheroids are not self-renewing stem cells, so they tend to be three-dimensional, but less recapitulating the actual structure and more of just, you can imagine, just a ball of cells, basically. Organs on chips, I will look at in a bit more detail in the next slide. And then I think you're all aware of the use of data. Certainly we're aware of computational modelling, and we've already heard a little bit about adverse outcome pathways. So we have lots of options when we want to look at non-animal methods for looking at respiratory disease research. And as I said, I'd like to focus a little bit on the organs on chips. This is Wikipedia's definition of the organs on chips. And I guess briefly, they are three-dimensional dynamic setups, something, I don't know, as big as a microscope slide, maybe with channels in it that we can put different cell types in and start to look at their interaction and their function. And this is Wikipedia said this at the bottom. So you won't be surprised to hear this from me as an animal advocate, but it's not actually my words. It's Wikipedia saying that one day organ on chips could perhaps abolish the need for animals in drug development and toxin testing, which is what I like to see. This is my favorite organ chip. The lung chip was developed by the Wyss Institute for Biologically Inspired Engineering, also in Boston. And I love this partly because it's lungs, but also because it won the UK Design of the Year award in 2015. So it kind of pulls together science and art quite beautifully. And it was on display at the New York City Museum of Modern Art for some time. And I'm based in the UK, so it's kind of difficult for me to see whether or not it's still there. But I'd love to know if any of you are anywhere near New York, whether or not that's still on display, because I think it's a fantastic, it's a thing of beauty. And it's functionally quite amazing as well, because it's been used or it's been developed beyond that kind of static culture that I was used to when I was developing airways models, to put a force across it that mimics breathing quite beautifully, so that we can stretch that lung exactly as if it was inhaling and exhaling. And that's pretty important if we're trying to recapitulate physiology more accurately. Then a lung breathes, that's what it's for. So we need to make sure that we're making it breathe. The video here, I can share the link if later, sorry, if that's useful, but that is a really lovely video that shows this lung chip breathing, and it shows what the functional implications of that breathing are. And it also shows the two features of this chip that I wanted to raise with you today is one, that we can look at immune cell recruitment using this. So we have lung cells on the top, the epithelium, and we have endothelial cells on the bottom, exactly as you would in your alveoli, basically, a very thin layer, really good for gas exchange. We can put blood cells in that capillary layer underneath, exactly as you would have in your body, and we can flow air or whatever we want on the top, again, exactly as you'd have in your body. So if we put neutrophils, so our innate immune cells, in that bottom blood channel, we can put them underflow so they can whiz around there. They're quite happy to do that. As soon as we add an inflammatory stimulus to the top, then the epithelial cells will send out the alarm, and those neutrophils will stop flowing happily. They'll stick to the endothelial cells, they'll climb through, they'll actually go onto the epithelial cells in that top channel, and they'll crawl about looking for the bacteria and phagocytose those bacteria. So exactly recapitulating what you would find in your lungs if you got an infection. And then the second way that this breathing lung chip has been used, which is quite amazing, is to look at a mechanism of edema in cancer patients who are taking IL-2. So IL-2 is a really useful therapy for people, but it tends to have a dose-limiting toxicity because it drives this pulmonary edema. And what the lung chip was used to show is that, number one, that breathing is required for this edema. So it's not related to immune cells, but it's actually something to do with the actual mechanical movement of breathing. And also gave us a suggestion as to what that mechanism might be. So they could use the lung chip to tease out what molecules are involved so that we can think about blocking them and allow people that need IL-2 for cancer to keep taking it beyond this dose-limiting toxicity. And if we look again, then if we come back to SARS-CoV-2, this was a group that actually from Don Ingber, who is at the Wyss and Harvard. So he has a position at the Wyss Institute, and it's from Harvard University as well, and is kind of king of the lung chip as I see it. But his group showed that during the pandemic, really, that animal models currently used as the benchmark for validating therapeutics, but when they're not natural hosts for these infections, they commonly exhibit different systemic clinical manifestations from humans. So it's really difficult in that situation where we need something quickly to look at developing an animal model that might have a similar symptom, but if that's a different mechanism, we're never going to get the right clinical intervention. So this is where the organ chip came into its own again, I feel, for drug repurposing. So this is using a fairly simplistic chip using just two cell types, so the epithelial cells and the endothelial cells. They looked at influenza A infection and also SARS-CoV-2 infection, and found that using a range of antiviral therapeutics that have been used in the clinic, that actually the chip beautifully recapitulated what the human clinical picture was. So those antivirals like the hydroxychloroquine that we kind of knew didn't do anything, it didn't do anything in the chip either, and they could show that it didn't block viral entry in the chip, whereas amidaioquine and its metabolite were capable of reducing viral entry. So as I understand it then, if you've got the amidaioquine, so the parent drug, and the metabolite, that maybe gives you options in terms of formulating these drugs. So the amidaioquine itself could be inhaled and it would have an effect because we showed that it would reduce viral entry. But the metabolite also suggests that you might be able to take it orally or systemically and still have some kind of functional use out of this drug. And it did go to clinical trial in South Africa, I think. But I'm not sure where that clinical trial went to. I feel like I looked it up and it hadn't done anything, or it didn't have any results posted, that's for sure. And I don't know whether or not that's because the vaccine appeared and suddenly we stopped being quite so desperate to look for drugs. But it's still, I think it showed me that there is a way that we can quickly look at what's already out there that's approved and see whether or not it will be useful. And this is another really lovely example, I think, of where the chips can be used for this, a fast response, I guess. And this I see this as kind of a perfect storm. So I'm not trying to overhype chips and saying they're brilliant for everything, because I know that there are not. There are barriers still to their use, and they're not, they're not perfect. But what we had here was a rare disease for which there are no animal models and also no current therapeutics. The chip here used human adult stem cells induced into neurons and Schwann cells to carry out myelination, because this is a demyelinating neuropathy. And what this chip was used to show was that if you put patient serum on this chip, you'll get fixation of antibodies, which leads to complement activation, which destroys the neurons. So we had a biological mechanism driven from the chip data that allowed the researchers, to say that actually we've already got a drug that's been approved that will block the complement serine protease, that's required for activation. So we had this biological mechanism that hopefully helps the regulators to see that their risk is reduced by putting this into clinical trial, and a pre-approved drug. So we already had safety data from this drug. So again, we reduced the risk a little bit, and that drug is now in clinical trial. And I can share the data from that after this as well, if that would be helpful for you. I didn't really want to bombard you with a lot of information on my slides. Maybe this will be more interesting for you, and you probably know all of this already, but we can look at NAMs for inhalation toxicity testing. And this work on the left came out a couple of years ago, and this is a new approach method for inhalation risk assessment that looks at fluid dynamics to have a look at deposition, and then uses a commercially available airways setup. So this MuCell model, which is a differentiated model of human airways epithelium that you can use to look at the barrier function of the airways and also cytotoxicity. And the OECD has used this as a case study to show that although this is not approved for regulatory use, as I understand it, it's kind of fit for purpose for regulatory use. And then I noticed last week that the US EPA released this model for human respiratory system. So looking at deposition. And it seems to me that the beauty of this is that you can change your deposition according to your gender or your age or your ethnicity, which is fantastic if we're looking for exposure of a population. We don't just want to use that kind of, one healthy white mouse kind of model. And this gives us that capacity to look to see where will your particle deposit, and do we need to worry about it or not? It's not just lung chips. So I was involved in a project with the European Commission, the Joint Research Centre, trying to create a snapshot of what advanced non-animal methods are out there for biomedical research. And I can share this if you're interested. It's not just for respiratory tract diseases. There were other groups that worked on cardiovascular disease and breast cancer and immuno-oncology. So there are other repositories out there if you're interested. And I think it's good that if we take a look at biomedical research models, because hopefully that's what's going to feed into regulatory at some point, or feed into the drug development pipeline. So we know that biomedical research is not thinking about validation or qualification. There's a lot of flexibility there, but it gives us a snapshot of what's happening and where might we be able to pluck what could be useful for looking more at safety testing, I guess. It's not just lungs. So lungs are my particular obsession, but actually, if we're thinking about drug safety, then liver chips are maybe more valid here, because we know that many drugs fail when they get into patients because of unexplained liver toxicity. And the liver chip, which is a commercially available chip that uses 4 different cell types, has been used in a really comprehensive study looking at 27 drugs, some of which were toxic in patients, by which I mean that they cleared preclinical testing in animals as safe, and then got into patients and suddenly got these toxic signals coming up that meant that they could no longer be used. So this study looked to see whether or not the liver chips would have flagged up that toxicity, and it found that actually the liver chip could distinguish toxic drugs from the less toxic structural analogues. And also, pretty importantly, I think the liver chip didn't falsely identify any non-toxic drugs as toxic. So the suggestion is that maybe we can insert the liver chip into that preclinical testing pipeline. So we do our simplistic, I guess, in vitro models as our first screen, then go into the human liver chip. And if something comes up that looks toxic, maybe we stop and we don't go into animals and we don't carry on, but we go back to our kind of screen and look for something else. And this study here actually looked at the economic analysis of this as well, and suggested that $3 billion could be saved if the liver chips were taken up for DILI, which has got to be attractive in everybody's world, hasn't it? And it's not just humans. So the same group actually earlier had done a liver chip using cells from rats, dogs, and humans, and looking at that concordance between different species. And what they showed was that there is concordance between the chip and the whole animal. So if something's toxic to the rat, then they would see the toxic signal in the rat chip. Now, we see there might be a couple of ways that we could use chips like this then. One is maybe trying to help our confidence that a chip recapitulates the whole organism. And maybe that's helpful if we are thinking about regulatory risk assessment and that decision making. Maybe we could look at which drugs failed in animals and see whether or not they have a safety signal in humans. Maybe that's a bit too risky, but it could be worth seeing what have we thrown out that maybe might be useful. And also we see that maybe using animal chips has a role in veterinary medicine. So perhaps we can reduce animal use for veterinary drug development by using the animal chips in the same sort of pathway or pipeline that we could use them in the human drug development too. And that is it from me. Thank you very much for your attention. And this is Teddy to say goodbye with his big ears.
Right, I think we have time for one or two clarifying questions. Joe, any online? No? Well, actually, Dr. Marshall, I do have one question for you, and it pertains to this liver chip at the end. And I'm kind of fascinated by the fact that it has been so good at identifying IDILI. And, you know, and again, this is not my area of expertise, but I do believe that a lot of the adverse effects in the liver tend to be mediated by the immune system. And quite honestly, the only immune component there are Kupffer cells. So I'm wondering how this is so effective. I don't know, maybe you could comment to it. Maybe the Kupffer cells are sufficient to mediate those responses, but you know, some of the literature suggests involvement of T cells and also B cells. So maybe you could just speak to that.
Yeah, I would put my hand up and say it's not my study, they're not my data, so I'm maybe not the best person to comment. And yes, I agree with you. I thought that was really interesting that this, I think, speaks to how complicated do we want the model to be. And maybe it's flagging up an element of that DILI, but maybe it's not the whole story. And if we put T cells in there, perhaps we'd see something additional that would give us more information as to why that idiosyncratic DILI happens in people. So it's not perfect. So it identified 7 out of every 8 toxic drugs. So maybe there's an element of that, you know, that missing toxicity is due to a more T cell mediated or more broader immune mediated response.
Thank you. And thank you for that presentation.